My name is Michelle Sabick, and I'm the president-elect of the American Society of Biomechanics for a few more hours today. And it is my great honor to introduce today the ASB Founders Award winner for 2019. The Founders Award is given by the Society to recognize scientific accomplishment in biomechanics and excellence in mentoring. It is open to investigators of all disciplines within the ASB. The requirements state the nominees must be current members of ASB who are between 5 and 20 years since receiving their terminal degree. This is a relatively new award that was presented for the first time in 2017 and was created to honor outstanding mid-career ASB members who are leaving their mark on the Society not only through their science, but also by their ability to develop and nurture the next generation of scientists. The award consists of an engraved plaque and a check for $1,000. The 2019 American Society of Biomechanics Founders Award goes to Dr. Silvia Salinas Blemker. Dr. Blemker is a professor in the departments of biomedical engineering, mechanical and aerospace engineering, orthopedic surgery, and ophthalmology at the University of Virginia. Professor Blemker completed her PhD in mechanical engineering at Stanford University in 2004. Prior to that, she received her bachelor's and master's degrees in biomedical engineering from Northwestern University and received an NSF graduate fellowship. She is widely known for developing sophisticated computational models that bridge molecular factors and muscle function. She has used those models along with state-of-the-art imaging techniques to address important clinical problems, such as volumetric muscle injury, cleft palate repair, and muscular dystrophy. One of Dr. Blemker's major research contributions to date has been to develop a mathematical and computational framework for multiscale modeling of muscle. Her nominators for this award noted that her 2005 paper in the Journal of Biomechanics was, quote, revolutionary, providing novel insights into the links between complex muscle architecture and mechanics. That same project earned her the Journal of Biomechanics Award in 2003, and she was also chosen as the Predoctoral Young Scientist Award winner by the ASB in 2004. Dr. Blemker's excellent scientific achievements have been balanced throughout her career by an outstanding track record of developing the scientists and postdocs that work in her lab. She and her research group have been awarded the Journal of Biomechanics Award in 2010 and were finalists for the award in 2017. Sounds like the beginning of a dynasty to me. One of her students won the ASB President's Award for Best Paper at the World Congress of Biomechanics in 2014. These are just a few of the many examples of the accolades that Dr. Blemker's group has earned under her tutelage. She has not only successfully mentored a number of postdoctoral fellows, PhD students, and master students, but she's highly committed to mentoring undergraduate students, and she mentions that on her CV, and she regularly maintains between 3 and 10 undergraduate students in the lab. When I spoke to Dr. Blemker's students, they enthusiastically offered to provide me with content for this introduction. They mentioned the quote that is on the top of her CV by Albert Einstein, which is, "Creativity is intelligence, having fun." And they said that Silvia embodies this quote in many ways. She encourages her students to think creatively about their own work, and she effectively models how to intelligently tackle important problems in biology and mechanics while still having fun. Students under her guidance have a chance to learn in a space where they have freedom to explore ideas and sometimes fail. I personally cannot think of a better definition for a supportive and nurturing mentoring environment. One of her PhD students told me, "Silvia will advocate when a student needs support, point out a new direction when they are stuck, and stand up to cheer, or maybe floss, for their successes." It is a privilege to have her as a mentor and friend. I hope we get to see a floss later. Finally, while it was not a strict criterion for this particular award, I want to mention another way that I have witnessed Dr. Blemker's positive influence on the broader scientific community. In 2018 and 2019, she was a Public Voices Fellow for the Op-Ed Project, and in March of this year wrote an opinion piece on women in STEM that appeared in USA Today. It was titled, "Astrophysicist Barbie is Perfect. That's not how you attract more girls to STEM careers." If you haven't read it, I encourage you to take a look. It was brought to my attention by a female engineering faculty at my institution who had found it inspirational and distributed it to all her female colleagues, including myself. I was proud at that moment to be able to say that I knew the author of the article, and I am proud at this moment to welcome Dr. Blemker to the stage to give us the Founders Award presentation.
Well, thank you for that introduction. It was really beautiful and humbling, and I love that the quotes. I don't know if I can floss, though. My husband sent me this article saying that moms shouldn't floss. So but thank you. It really, truly is an honor to be up here, to be able to receive the award and give this presentation. I really grew up in this society. My first presentations were in this society. Many of the friends that I've made over the years is as a result of being a part of ASB. So it's really a place I call home, and I'm excited to be here. The title of my talk, I think, will become clear as it goes along. If not, then sorry. But that was a rainbow that we saw in Canmore I guess it was like a week ago. I love rainbows. I think my students will tell you that. There's often rainbows all over the lab. I just love color, and I love creativity, and I love just that it's something that emerges from rain and clouds. And so really part of the inspiration for this talk actually really came when the idea for it has been brewing, but I really came true to home when I went to the mentorship session early in the meeting. I don't know if many of you went to it, but it was fantastic. And one of the big pieces of advice that the group gave, phenomenal group here as you can see, is to really be you. A way to succeed in a long career of research and academia is to be yourself. That's where we're comfortable, that's where we can find joy in what we do. And so, and that's something that I totally agree with, and I think over time I've worked really hard to be that. And so here I am today giving a little bit of a weird talk about rainbows. But I also have been thinking a lot about why it's hard. Like, why is that advice that we even have to give? Why is that not obvious? And that's because, as reflected here, I've put some of my my esteemed colleagues here with me. We all are unique, right? We all kind of, if we parallel who we are with a shape and a color, we all come in lots of different colors and shapes, right? And that makes us do really interesting research. It makes us tackle interesting research problems and makes us view things uniquely. However, professional advancement usually requires us to go through hoops. I don't know if, any of us, pretty much all of us in the room know what I'm talking about, right? Hoops, get through a undergrad degree, go to a PhD, and these hoops are relatively rigid. If you think about what it takes to be a successful PhD, I think we are relatively, and overall as a system, rigid. What's a successful postdoc? How many papers? What types of presentations do you give? What's your publication record? How many grant funds do you have? What are your awards? How do you define getting awards? All those things are relatively structured, I would argue. And so when we are each unique individuals with our own shape and size, for example, here me as a rectangle. I don't know if rectangle was the shape I would choose, but it was the easiest to model here. So I'm the rectangle. And so if you think about how to succeed and get through these hoops, if you look at the rectangle, it doesn't fit perfectly in the square, right? So what are the options for a person, let's say me? Well, I could decide, well, that hoop, it's not a good fit, I should do something else. And I think many of us know that we've seen many students like that, who think that they're not the right person for research or academia, and so they decide to do something else because they see the hoops and they don't think they fit. The other option, which I think most of us do to some degree, and it's not entirely bad, is we squeeze. We get ourselves through the hoop. Get like really stretchy in weird ways. And I didn't actually, it's funny because I didn't plot, this is like a pseudo simulation where you can imagine the red parts are getting really highly strained or compressed. Maybe I tried to stretch a little bit in the other direction, a little Poisson's effect. And stress is here maybe mechanical in nature, but it's actually real, right? When you're trying to fit yourself into a mold of some kind, a hoop, that can be stressful. and cause tension in your being, in your personal life, in your career life. And ultimately, what happens afterwards is I would argue that we have residual plastic deformations, or residual stresses, that lead us to change our shape a little bit. And maybe that's not entirely bad, we grow, right? But over time, as you filter that, then what you're doing is maybe after you go through many, many hoops, maybe our system is whatever the people who get through the hoops start to look similar, right? Which is not eventually what we want. We want us all to be distinct and unique. So in this talk, I wanted to talk about a few things, and I'm going to weave research in here in odd ways, so bear with me. I wanted to talk about two things. As myself, what have been my strategies, and really many of those I've learned from my peers and my mentors, about how to be elastic, how you get through the hoop and you sort of bounce back and be as close to who you are as you can. And the other one is more broadly, how as a field can we make our hoops more elastic? How can we promote people to get through these hoops that we know need to exist, right? We need to have standards, but how can we make them flexible so that many shapes and sizes, they appear to be, we don't filter people out because it looks too rigid, but also people can stay who they are and don't have these residual stresses. As I move on, I wanted to talk about a few things that I wanted to talk about what makes you, you, or me, or me, you, not me, you. Sorry. So of course we all have our own personal story, and that story keeps evolving as we have a long academic career. And one of the things I love about being an academic and going to conferences is that we are friends, and so we also share our personal stories in our personal journey together. We all have our educational background, we have our knowledge and skill set, which of course continues to evolve. We have our viewpoint, and I have my handy lens. For those of you at ASB last year, you know what this is. We have our network of people that grows, and we have our goals that also change, but we want to keep our own personal career application goals in mind, and we have our ideas. So just a couple things about what I would say, what makes me me, just giving you context. A little bit about my personal story. My parents are immigrants. They came to the U.S. from Argentina before I was born in 1968. They came. I was born after that. I have an older brother. I think that makes me like a younger sibling. In many ways, that's part of my personality. My father was blind, so I grew up with a disabled parent. He was fully blind, but he was a mathematics professor, and he passed away when I was in right after I finished grad school. My mother was a professor too, so partly why maybe having professor parents makes you kind of unique. I grew up in Lawrence, Kansas. I'm married with 2 children. That's a picture of us from last October. And as you can imagine, my husband is probably sillier than I am, and as are my children, Mario and the color orange. And another part of my personal story that is very relevant here is that my younger son was born with medical problems that made his first year of life very challenging. And I did have him before I went through tenure. So that is an important part of my journey that it's going to come back later. So, okay, now let's move on to the kind of the part that determined my research career. So, probably the first person who really had a big influence on me is Scott, who I think I saw him back there. There he is. I was an undergrad at Northwestern and took his class and got inspired. I ended up doing my master's and PhD with him, and at that time, so as you all know, open SIM models have become ubiquitous. I always like to be the weird one, and I looked at those and said, well, they're lines, but muscles are 3D. So decided to move into creating 3D models of muscle. And Scott really empowered me to think differently, develop a new skill set, and do something different. And that's something I carry on with me as I mentor other students. That's something that has really impacted me. So that was the beginning of my world, was creating new ways to model muscle. So then I started my faculty job, and I don't know if you guys all felt this, but I remember the first day of my faculty job, I sat down at my desk and I was like, now what? What am I supposed to do? It was a clean desk, and nobody was around. I'm like, okay. So looking on to where I was, I did have a knowledge and skill set, and I did have a lot of ideas for where to go. So one of the first things I did was recruit a student who actually knocked on my door that first day and said, do you need a student? A woman named Bahar Sharafi, and she chose to tackle one of the things I was excited about, which was also the hardest project, was moving from modeling muscles in 3 dimensions, tissue mechanics, to looking at the microscale. That was something I thought would be really cool. I had no idea how to do it. She didn't have any biology background, but she's a brilliant mathematician. And so together we did that. So that was exciting, developing new tools. The other thing that happened around that same time was actually at an ISB, ASB, I believe, in Cleveland. Was there one in Cleveland? Yes. I was chatting with Daryl Thalen, and he was at Wisconsin at the time, and he said, hey, Sil, why do you think this muscle's always injured in the same place? It seems like you might be able to figure that out with your models. And at the time, I was like, I have no idea. Well, it turned out that we started talking more in depth, and we talked about the questions of are the injuries where they're localized due to high localized strains? That's something I can predict with my models. If so, could then the 3D muscle morphology explain why these muscles are injured in that region? And also even explain why certain muscles are more injured than others. So we ended up, it was a really exciting moment for me, because I still collaborate with Daryl. Is he here? There he is, yay. Has been a mentor for me and a collaborator for a long time. So we started by, we ended up getting some NIH funding, which was awesome, but and surprising for me. But so we ended up creating some 3D models. A new student, Mike Rehorn, came along and built these 3D models that predicted strain distributions throughout the biceps femoris long head. And indeed, if you look here, so we created models from MRI, which is much similar to what we do now. Of course, it took him a lot longer than it used to, or it took him a lot longer than it does now, but we predicted a long fiber stretch within the muscle, and this is the proximal muscle tendon junction, and this is precisely where the muscle commonly gets injured. So indeed, the 3D models could give insight into this injury question, this set of injury questions. And Mike went on to figure out, well, what aspects of the muscle give rise to these localized strains, i.e. what about the muscle makes it prone to injury? And it turns out it was a difference in aponeurosis dimensions that gave rise to high localized strains near this proximal muscle tendon junction. It was pretty exciting, actually. A fair number of people in the field caught on to the idea, and it's been neat to see where that went. We also, at the same time, I recruited a student, Nick Fiorentino, who's now at the University of Vermont. His PhD was to do the very hard task of developing, applying imaging techniques to validate the model prediction using cine-dense MRI, where he built models based on image data and compared the strains within the muscle in both passive conditions and active conditions between the model and experiment, which allowed us to gain a lot of confidence in our predictions as well. And really, while I had done a fair amount of imaging as a grad student, I really was a card-carrying modeler. So a lot of the experimental work that I do now in the lab is really thanks to students willing to teach themselves a lot and teach me along the way. The other thing that Nick did, which was cool, is he realized that there was a fair amount of variability across subjects in terms of their aponeurosis dimensions. So some people had a very wide or a narrow proximal aponeurosis, and I was saying that the difference between these aponeuroses is the reason why we get high strains. So some people have narrow proximal aponeurosis and some people have relatively wide proximal aponeurosis. So then he compared the principal strains near that region between the subjects that have a narrow aponeurosis and subjects that have a wide, and realized there's a fair amount of difference, which is, for me, exciting, showing that the internal morphology of the muscle and the aponeuroses actually can be an indicator for an injury susceptibility, why one person might be more susceptible to injury than others, which gives you some more clinical applications. So there's more work that came out after that, but one thing I wanted to bring up, now thinking about as we were finishing that research and kind of moving on, I did spend some time figuring out what I wanted to do next and where I was going, and realizing that it was an exciting work, but I also always really wanted to get back to studying diseases, especially diseases that affect kids. I spent my entire PhD working in a lab that focused a lot on cerebral palsy and improving outcomes of treatment for kids with CP, and I'd done imaging studies with my colleague Deanna Asakawa, and really wanted to get back there. And so I had that in my mind, like where can we go? And at the same time, as I was doing research, reading about muscle injury, this paper and many like it kept coming up. And I don't know if this happens to you sometimes, but usually when it's like words that I don't really know, I'm like, it must be irrelevant. Like that paper just must not have to do. It must just be an error in the search thing. And eventually I just was like maybe bored one day or something, and I just decided to click on it and read about it. Like, what is this? Like, I kind of knew what dystrophin was and I knew what the sarcolemma, but I just didn't know what this was about. So I started reading it, and it turns out that in Duchenne muscular dystrophy, you lack the dystrophin protein that connects the cytoskeleton to the fiber membrane, and which connects to the ECM. And it's the absence of the dystrophin protein that leads to Duchenne muscular dystrophy. And what happens then is boys with Duchenne muscular dystrophy start quite with healthy looking muscles, but as they start moving, they're actually injuring their muscles over and over and over again. And that's what leads to the progressive quick wasting of their muscles. So over time, they have changes in their walking pattern. They don't develop typically. And if you look inside their muscles, fibers are being replaced with fibrosis. And you see all the hallmarks of injury. You see inflammatory cells, you see damaged material. So these muscles are very susceptible to injury. And that's why I was like, oh, that's why that keeps coming up, because I'm typing muscle injury, and this is muscle injury. So reading more, realizing that this is a devastating disease. Boys with DMD end up in a wheelchair by the time they're between 12 and 16 years old. At this point, they live maybe to their late 20s to early 30s, but there's really there's no cure at this point. Treatment is really palliative. It's really to improve the quality of life. So so it's a pretty, and I'm now connected with the muscular dystrophy clinic at my institution, and have met patients and families, and it's really devastating. So it's a place that we can make a difference. I thought, well, I modeled muscle injury for that application, maybe I could do this. So, and at the same time, back to Bahar, she had been developing a lot of these models further, and she had some new ideas about how to model strains within a fiber, and even model dystrophin. She had been kind of listening and had her own ideas and been reading other papers, really thinking the way she does. And so we expanded on her models and looked at how these are models of muscle fibers in the extracellular matrix, and she looked at how tissue properties varied between healthy and dystrophic if you take out the connection between fibers and the ECM. She also looked at how fibers strains are localized within fibers if you have a dystrophic versus a healthy, showing that for the same lengthening or eccentric contraction, you're going to get more strain in the fiber, which could explain why these muscles are more damaged. So we wrote a grant. That was in 2009. I started in 2006, so it was pretty junior. And as you can imagine, it didn't go great because it was pretty outside the box. I was not from the field of muscular dystrophy, and my modeling techniques were different in different ways. Maybe I probably made some bad grant flaws. But I got some interesting feedback. One of them was, well, you're modeling the mechanics, but you haven't addressed the issue of degeneration and regeneration of the muscle. And that's really the problem. That's a huge part of the disease progression, and all you're doing is the mechanics. And I was like, oh, darny, but the mechanics was hard. So it's like multi-scale and all these things. What do they want from me? So anyway, but I got that comment. And then the other one, which speaks to this, the issue of the hoops. 3 years into my faculty job, I got the comment, a significant concern is the relatively low productivity of the PI since arriving at UVA. And that was like daggers. Oops, whoa, that was loud too. That was pretty hard to take. You know, maybe they had a point, but I was like, I just started, I already had a baby, like, I don't know. That was that. So then here are my options. Ditch the ID altogether, like shove it, like maybe this is too far out, moving into an entirely new clinical problem. Maybe I shouldn't do that. Maybe I should stay within the area that people know me and you know, move on. Or maybe I just I need to adjust my shape, maybe develop myself further, and maybe allow time for the hoop to change too. Sometimes I do think our ideas, especially when we're thinking differently, are ahead of their time. It's just not the right time for them. And we just have to wait a little bit till the hoop adjusts to where we're thinking. And then indeed, a few years ago, I did receive a UO1 grant with the title Multi-Scale Modeling of Treatment Discovery in Duchenne Muscular Dystrophy. So a lot of stuff happened in between, and now that's a big project in the lab. And it was almost something that I shoved under the rug. But and I wanted to tell you a little bit about how I was allowed to changing my shape to make it fit the new hoop, so to speak. So in addition to studying how, modeling how the muscles are susceptible to injury, I also realized that as a result, the muscles are susceptible to chronic inflammation. So because the muscles are being injured all the time, there's a bunch of inflammatory cells, there's lots of stuff going on, and that leads to what's called a profibrotic state. So now we know the muscles are fibrotic, but then they turn into this like vicious cycle of generating more and more fibrotic tissue. And even the stem cells within in the muscles, the satellite stem cells, the ones that are responsible for regeneration, they also are changed in DMD. They actually are responsive to the lack of the dystrophin gene. So that's, and like I understand that comment now. The other thing that happened was a colleague of mine who actually is also one of my great friends. We shared a nanny for a long period of time, and over time I just kept listening to what she was doing. And she, her name is Shane Pierce-Kotler, and she is an expert in something called agent-based modeling that simulates the behaviors and interactions of cells to predict emergent responses of tissues. And the types of responses that she was normally simulating was growth of the microcirculation, angiogenesis, changes in the microcirculatory system. And I just remember one day talking with our babies saying, well, can you simulate like the changes in the muscles, in the muscle too, you think? Like, so she taught me about agent-based modeling. So here's how it works. So you have a cell, a simulated cell called a fibroblast, or here we're simulating a fibroblast, which is the cell in the muscle that creates the ECM, or is the kind of responsible one for the fibrosis. And let's say there's different types of factors, biochemical factors around it. So it's like you can simulate the biochemical environment within the muscle. And you can simulate behaviors such as proliferation, so dividing, migration, so maybe we know that a fibroblast likes to be around a certain type of growth factor, so it'll tend to move over there. It walks a little bit. You can model how it differentiates into different cells, maybe changes its state to being very secreting of collagen. It can secrete other factors, and it can die also. And so these are the types of behaviors that you can model for each individual cell. And all of these you derive from experimental observations. So a student that's working on a thing like this, so Cal Martin was the first student in our lab, who's the one that won the ASB President's Award. He was the one who developed the first agent-based model of muscle in our lab. And he got a very in-depth understanding of the literature as a result, because he had to program all those in. Then another student named Kelly Virgilio took the original model that Kyle built that was primarily for predicting muscle atrophy from disuse. I love this picture of her because she was very pregnant, giving a talk, also managing experiments. Anyway, so it's a great picture. So she developed the model into something where we could prescribe an injury, we had been studying what leads to the injury in DMD, and then model how the tissue responds. So the injury is the blue stuff, that's the bad stuff, the necrosis, and then we can model how the muscle adapts, shrinks, and then regrows as a result of the injury. And so then we can model all the different cell responses in the muscle, and also compare with the literature. So this gave us, so by including this concept into our conceptual framework, that really allows us to link the mechanics with the biology and really predict what drives the regeneration of the muscle and start looking at the interaction between factors. Mechanics and inflammation is a big one. So that was really exciting. And now in the lab, you'll see lots of interesting things happening. Xiao Hu is looking at how muscle loading, what it looks like in boys with DMD and how that differs with the mouse model of DMD. Adrian Williams is building a 3D model to understand how the strain distributions within a muscle might influence distribution of damage within a muscle. And Emily Miller is a recent MS student who advanced our what we call micromechanical models to allow us to predict damage within the muscle, which we're comparing with experimental measurements of damage as measured by kind of traditional assays in the field. To couple that, we're also doing a bunch of experiments across scales. Brian Jones, a postdoc, worked with Kelly and developed a new way to test passive mechanical properties of dystrophic skeletal muscle. Hunter Wallace is in the room. He's developing novel biaxial testing measurements of the diaphragm muscle, which is in particular very damaged in boys with DMD. Ultimately is often the reason why they die is due to respiratory failure. And Riddhi Sahini is also in the crowd. She's looking at the real microstructure of the ECM. Through our modeling and experiment endeavors, we've realized that fibrosis in ECM is very complicated, number one, and it looks very different than we thought it did. So this is all exciting stuff. So I'm kind of going superficial here, but really showing you how some initial ideas and finite element model can lead to lots of different directions, and really us kind of conforming or kind of expanding our shape to fill the hoops necessary to grow into this field that's new to us and new to our field, the field of biomechanics. The next thing I wanted to talk about a little bit is personal goals as they relate to career goals. So I mentioned before that I had a son who had medical issues, Daniel. He was born in 2010. He was born with something called Pierre-Robin sequence, which I hadn't heard of. So literally my husband and I were Wikipediaing it right after we heard. And so it turned out that with that comes, he had a small mandible, which leads to difficulty with breathing. And so actually a common treatment for that is jaw distraction, which, do you guys remember that Dennis Carter's group was studying jaw distraction, right? I was like, oh yeah, the thing with the things. Fortunately, my son didn't need that. But he also had a cleft palate, which I knew nothing about too, which meant he couldn't eat very well. So an infant who can't breathe or eat is pretty challenging, as you can imagine. Fortunately, that passed after surgery and whatnot, but I wanted to learn a little bit about cleft palate. So it's pretty problematic, and it's different from a cleft lip. So a cleft lip is a failure of the alveolar ridge, or the front of the lips, to close. But the cleft palate is when the roof of the mouth doesn't close in utero, and so there's a hole there, and it's pretty important because you need that for eating and speaking. And so children that have a cleft palate have a surgery done, but it turns out the surgery isn't very successful. It's pretty often that the outcomes after a while, after surgeries are done on the babies when they're about 10 months old, and about 5 years later they start to assess the speech, and you have a lot of kids who don't speak very well. And so the big moment for me came when I realized that there's actually a whole bunch of muscles under there, in the soft palate. So if you take your tongue and touch the roof of your mouth, it's hard, but if you were able to go further back, you would feel a soft thing. And it turns out that soft thing has to move up and down the soft palate while you speak in order to make the sounds of the English language. So that's the muscles back here. And there's a primary muscle called the levator belli palatini muscle that pulls the soft palate up and back as you speak. And in order to make most of the sounds in the English language, we need to be able to do that. So in a child with a cleft, because there's a hole there, that muscle is misdirected or misaligned. And the goal of the surgery is to realign it to allow it to flex and move. During this, after the surgery, a lot of kids, instead of being able to form this closure with the muscle, so we're looking at a cross section here, the muscle's going in and out of the page, it contracts and pulls the soft palate up into the back of the throat, but they aren't able to achieve that closure, and that's called velopharyngeal insufficiency. And here I'm going to play a video. Let's see if it works. So that's actually Daniel when he was about 4, so he's cute, isn't he? But as you can sound, it sounds pretty nasal, right? His S's were almost non-existent, and a lot of the rest of the sounds sounded pretty nasal. So that's a pretty typical sound. Some of that can be improved with speech therapy, but often children require more surgery to fix this. So I decided, well, that's muscles and it's surgery. That's what I know about. So I'm going to bring personal and career together. I'm going to fast forward a little bit. This is a big project in the lab. I wanted to acknowledge 3 students, Katie, V, and Josh, who have all been working hard on this problem, building 3D models of all the muscles of the soft palate from MRI, doing dynamic MRI, and doing things like driving the activation of the muscles that lead to shortening the muscles and forming closure. We can do things like measure the closure force, and so in concept, a child that has a cleft won't be able even to make closure force, or maybe they can make a little bit of force, but it's really difficult, so it's hard for them to talk. We can look at things like muscle shortening, closure force. So just shoving it in there, but there's a lot more work there to talk about, but just wanted to talk about this research arc, so to speak, and how the personal kind of changed my shape, but then how the professional shape changed, and it kind of all worked out. So it was really realizing my skill set had a lot to offer after reading and talking about the collaboration. So I'd started collaborating with my son's surgeon. I also found a funding source that was willing to take a chance and fund a project that I had literally no credibility in. So it definitely wouldn't have worked for the NIH. Actually, the Hartwell's mission is to fund things that the NIH wouldn't fund, which is really empowering. That allows us to adapt into new fields. So if I hadn't found then, I wouldn't have been able to hire these excellent students, Josh, Katie, and V, to work on the project. I also, as a result of doing that and presenting some of our initial work, we actually were able to connect with some thought leaders in the field. And even today, this surgeon, Al Wu, actually has been presenting a surgery that is consistent with some of our model predictions, and actually some surgeons are adopting the new way that we've predicted how to do the surgery based on oral modeling and these collaborations. So we've seen changes in surgical practice, and now it's a highly active part of my lab. It's something we do every day, we talk about. So lastly, my last example, I promise, is having done that gave me a new lens on muscles, literally and figuratively and everything else, which I also was thinking about. The lens is like the way that we view the world, right? And the lens is sort of like a tree trunk. Over time, our experiences are going to influence our lens, and hopefully, you can imagine that as we get more and more focused, it would make it a little bit more and more cloudy and very centered, but hopefully some of the things that we can experience can actually make our lens more clear and more able to see different things. But Katie Knaus, who's here, Blemker, and she's presented a lot of work on the plantar flexor. So I wanted to tell you something about the other thing that she's done in her PhD, which is impressive, is demonstrate that you can model any muscle with our techniques, kind of inspired by the speech muscles. So it's a collaboration with a company called ASB, where we've aimed to model the ciliary muscles of the eye, which is the muscle that controls accommodation. So accommodation is when we look from near to far. And as we age, we lose that ability, right? But it's actually a muscle that does that. Some people think it's that inability to make focus is changes in the lens properties that get stiff. Well, a lot of it actually has to do with muscle contraction. So Katie aimed to do this. So she's developed this is her finite element model of the eye, the anterior eye. Here's the lens, here's the muscle, here's the outside of the eye, and these are some things called zonules that are like miniature tendon-ish things that are really complicated. And these are her ciliary muscles that have different properties. She's validated with a number of experimental measurements and actually used her model to help this company design some of their clinical trials. And one of the things that, so this is a simulation going from a relaxed to contracted muscle, comparing the function of the muscle, how contraction of the muscle, which she's measuring here, is changes in the shape of the muscle and thickening with lens thickening, which is going to change the focal point, and comparing that with experiments. But one of the things, the lens things that I thought was wanted to share with is now we're thinking about how this muscle, the mechanical advantage of this muscle to deform the lens, which is sort of like a moment arm, right? Mechanical advantage, moment arm determines the relationship between force and torque and then movement, of course. So Katie came up with this neat way to relate muscle length change with deformation, and that's what we're calling mechanical effectiveness. We didn't want to call it a moment arm because that's not a moment arm, but it's change in length of the muscle as it relates to change in thickness of the lens, and how we can measure each different region of the fibers. There's these three regions, and how they're actually antagonists to each other as it relates to the lens changes, but also they have opposing actions when you look at how the lens moves, which actually, when we've shown this to our collaborators and other scientists, it's actually given them a whole new viewpoint on how this muscle works, really putting it in the language that a biomechanist has, which really illustrates how, like, if you kind of think outside the box a little bit and you realize that actually the concepts we have here can really be applied in ways that, you know, we hadn't really thought about. All right, so final words. Back to this whole idea about elastic hoops and whatnot. So strategies for being elastic as you get through these hoops. Always remind yourself of who you are and realize that's changing, and be patient. Some of your ideas are ahead of their time. Also, surround yourself by people who value you, who help you remind you who you are and why you're doing what you're doing. And also keep growing as you want to, and then see opportunities where you can make a difference. And what can we do as a field to help these hoops become more conformable? Value different types of contributions. Understand that one timeline doesn't fit all, like expecting a bunch of papers after 3 years, you know, maybe not knowing I had had a baby and whatnot, but realizing that time is just a thing. It's not the end all be all. Reward and promote diverse applications. Give plenty of opportunities for people to reinvent themselves. As a society, you know, we could have an award or a grant mechanism for people applying for things that are totally outside of their main regime. Help them reinvent themselves. They almost have to have no papers in the area in order to get that funding. So as a kind of result, at the end I wanted to bring back the rainbows and show you and just talk about, you know, flexibility will have a huge positive impact. I think our field as a whole is very flexible and nurturing, but I think we all agree we have places where we can grow in terms of, and this will lead to promotion and retention of more diverse individuals, growing our field, broadening the applications of what we do, making our field more relevant to society, adding new things we do, and impacting more lives. So with that, I'm going to attempt to acknowledge, I acknowledge a lot of the people on the way, but really the research that I do is a joy because of the students I work with. It's all their ideas that are here, and it's just such a delight when they come in my office and show me a new result that they came up with. It's like, it's really the best thing ever. And our funding sources, of course, and collaborators and friends, and that we do have fun doing science. Thank you.
I think I went wrong. I don't have to.
So with that, I'd like to officially recognize not only a fantastic talk, but Dr. Blemker as the 2019 award winner of the Founders Award from the American Society of Biomechanics.
Congratulations. Thank you.
Right, before everyone leaves, I have one tiny bit of housekeeping also that I was instructed to provide. All running and wearable running mechanics sessions for today that were scheduled in McLeod E4 are going to be moved to Glen 208 and 209. And the prosthesis and arthroplasty sessions for today were scheduled in Glen 208 and 209 are going to be moved to McLeod E4. So those two sessions have been flipped. I'm told that if you update and refresh your app that the correct room numbers will be in there, but just please plan accordingly if you were planning to attend either of those sessions. And thanks to all of you again for attending, and thanks for being here so early on a Sunday morning and being available to hear this great talk, and enjoy the rest of your day.